said in the last video, we're treating some questions taken from different examination bodies like West African Examination Council, we have the Joint Matriculation Examination Board, and we have other sources of question. So today in this video, we still go through some series of questions from different topics, basically on what we know as heredity. So heredity has been a concept that is very taxing for students and teachers in schools. So that's why we are taking time to just break down what it means to understand heredity and how to approach questions in heredity. So I believe you're with your writing materials as we go on solving questions on today's video. So let's start with our question. So the first question we have on our list is, on what structures are the units of inheritance situated? On what structure are the units of inheritance situated? So we have the first one, which is the Golgi bodies, and we have the second one, which is the ribosome, and we have the third one, which is the chromosomes, and we have the fourth one, which is the endoplasmic reticulum. So we are being asked that on what structures are the units of inheritance situated. So for us to identify where the units of inheritance are being situated, we have to know what a cell is, and we know that in a cell, like you can see what I put on the board, you can see this is an average structure of a cell. So a cell has a central, basically this is an animal cell, so it has a central organelle, which is a membrane bound organelle, which is the nucleus. We have the nucleus, and inside the nucleus we have structures that we call the chromosomes. Chromosomes are spindle-like structures that are inside the nucleus. So, and on these chromosomes are what the units of inheritance, which are the genes, are located. So the genes are located on the spindle-like structure, which is the chromosome. The chromosome is found in the nucleus. We have the chromosome in animal cell and plant cell. So back to our question. So the structures on which the units of inheritance are situated are our chromosomes. So we have chromosome. Chromosome is a structure on which the units of inheritance, units are situated. So, and mind you, these units of inheritance are the one responsible for the development, are the one responsible for everything that characterizes an individual. Talk about your shape of the eyes, your shape of the hair. It's as a result of the combination of this chromosome. That's why we are able to have a high color to be like we have the shape of the hair and the mouth and all that makes up an individual. So that's on our first question today. So to the next question we have for the day, we are asked if a DNA strand has a base sequence CCA. So if a DNA strand has a base sequence TCA, its complementary strand must be, what's a DNA? DNA, we know DNA is known as deoxyribonucleic acid, and it is responsible for genetic transmission. So, and a DNA is a double helix structure. DNA is a double helix structure which has another strand. It has two strands, which the first strand being the major strand and the other strand being the complementary strand. So let's look at what DNA is made of. DNA comprises of some major nitrogenous bases, which are 4. I'll rub the board. So we have 4 nitrogenous bases that are made composition of the DNA, which are our what? Our adenine. We have our thymine, we have our cytosine, or cytosine as some people will say, and we have the last one, which is the guanine. So all these are the bases that are found in the DNA. So, and to take notes, a DNA has thymine as its base compared to an RNA strand. An RNA has what we know as what? Uracil. Uracil is the last part that is substituting for thymine in a DNA strand. So back to what we are saying, our adenine is represented by a letter A. So our thymine is represented by a letter T, and our cytosine is represented by a letter C, and our guanine is represented by what? Letter G. So these are the strands in a DNA. A for the adenine, C for the thymine, and C for the cytosine, and G for the guanine. So looking at all these strands, these are the basic strand, and we are given a question that if the strand of a base sequence is TCA, I'll rub the board. If the strand of a DNA is TCA, and mind you, our T is representing our thymine, our C is representing our cytosine, and our A is representing our adenine. So this is a strand. This is a strand for a DNA sequence. They're now asking us for the complementary strand of this. And in a DNA, mind you, before I go into how we get the complementary strand, you should note that adenine always bond with what we know as thymine. So we have a bond between adenine and thymine, and we have a bond between guanine, we have cytosine, or cytosine rather, bonding with our guanine. So look, with what we have on the board here, we have our G bonding with a triple bond to C, and we have our A, which is the adenine, bonding with a double bond. This is a double bond, is bonding with a double bond to T. So A bonds to T. So back to what we are asked, the base sequence that we asked to find the complementary strand is TCA. And since we know that our T will always join to A, which is our adenine, so we have the complementary strand of T to be A. And since our C will always bond with G, which is our guanine, we have our C to have the complementary strand of G. And since we said our A, adenine, will always bind with thymine, so we have our A to have a complementary strand of T. So the new strand that we have in this DNA sequence is what we know as what? AGT. So we have the strand AGT as a complementary base sequence of the DNA. So the correct option, the correct answer for this is AGT, as you can see on the board. Please, mind you, thymine is replaced in a RNA strand with uracil. Thymine is replaced in an RNA strand with uracil. Alright, let's move to our next question. Back to our next question. Homologous pairs of chromosome separate during which process? Homologous pairs of chromosome. We are asked the homologous pairs of chromosome separate during which of these? We have our option A to be meiosis, and we have our option B to be cytolysis, we have our option C to be mitosis, and we have our D to be cleavage. Our rubber. So I hope you following what I'm saying. These questions are what can disorganize students in any examination condition. So homologous pairs of chromosome, mind you, anything that is called pair occurs in two. Pair, when you say for example, you can see a man or a woman with a pair of trousers. A pair of trousers is not just a single or part of the trouser, but a pair of trousers is two. You can see a pair is just denoting two things, and the homologous pair of chromosome. So for homologous pairs of chromosome, let's represent a chromosome to have a structure like this. So these are chromosome. A chromosome with a center called what? Centromere. And we have what? The chromatids. This is the chromatids, this is the centromere, and we know this, as drawn on the board, is called what? Chromosome. So this is a chromosome, but we have what we call homologous pairs of chromosome. What's the meaning of homologous or homolog? Homologs are the same pair. When you say homologs means something that is of the same kind. So we have homologous pair of chromosome. That means we have the same pair of this chromosome. So with this structure, we can say these two chromosomes are called homologous pair. So homologous pair, this is the homologous pair of chromosome, and homologous pair of chromosome separates during which the first option we have in the question is meiosis, and the second option is cytolysis, which is option B, and this third option is mitosis, and the fourth one is cleavage. So I'll take the options and tell you the meaning of all this to arrive at the right answer. Cleavage. What is cleavage? Cleavage means a division or successive division of a cell. Cleavage is a division that occurs in a fertilized cell. So cleavage is a series of division that occurs. So having known that cleavage is a series of division, let's go to mitosis. Mitosis is a division that occurs in a somatic cell. Mitosis is a division that occurs in a somatic cell, and it occurs with a chromosome. So you can attach your mitosis to what we know as what? A single chromosome. In mitosis, we have a single chromosome separating. So back to the next option, which is cytolysis. What is cytolysis? Lysis, from the word, you can break this word into two: cyto and lysis. Cyto from cytoplasm and lysis from word breaking. So this is the breaking of the cytoplasm. The cytolysis is the breaking of the cytoplasm, and which will not be the right option. But going to the first option, which is called meiosis. In meiosis, homologous chromosome separate. We have homologous pair of chromosome coming together in what we know as melting division. So a melting division is characterized by what we know as what? Homologous pair of chromosome. Note the word homologous pair. In melting division, any melting division, you have homologous pairs of chromosome coming together, and they separate at a stage that we know as what? Anaphase stage. They separate at a stage known as anaphase. I'm very sure when you continue with this video, you will see and you get a proper understanding of what we mean by all the stages of mitosis and meiosis, we'll be able to break down everything that you need to know as regards the concept of meiosis and mitosis. So for our answer, we have meiosis. Homologous pairs of chromosomes separate during the process meiosis. Alright, so I hope you've seen what's on the board. So back to the next question. I'll rub. So we have the next question to be, which type of cell is produced by meiosis? Which types of cell is produced by meiosis? Meiotic division. We've talked about meiotic division being occurring with a homologous pair of chromosomes. They're now asking us which type of cell is produced by meiosis. The first one we have a fertilized egg cell, and the second option we have what we leaf cell, and in the third option we have what we know as red blood cell, and the last option we have what we know as what sperm cell. So they're asking us a question that in which type of cell does meiosis occur, or is meiosis undergo? But let's first of all look at the first option. The first option is a fertilized egg cell. What do you mean by a fertilized egg cell? A fertilized egg cell is the fusion of the male and female gametes. The fusion of the male and female gametes give us what we know as a zygote. So a zygote is formed as a result of the fusion of the male gamete and the female gamete, and the male gamete is called the sperm cell. The male gamete is called the sperm cell, and sperm cell has what we know as what haploid number of chromosome. Haploid number of chromosome is represented by N, and we have the female cell, which is the ova. In the plural form, in the singular form, we have ovum. The ovum also have what we call a haploid number of gametes, which is N, represented by N. So when we have the fusion of this N and N, we have what we call a 2N, resulting back into what we call mitosis. So a fertilized egg cell is as a result of what? Mitotic division. So we have a mitotic process in a fertilized egg cell. A leaf cell also is a somatic cell. And what do we mean by a somatic cell? A somatic cell undergoes mitosis. A somatic cell is a cell that will divide into the same, to give the same offspring as a result of mitotic division. So our leaf cell undergoes what known as mitosis. So in mitosis we have the cell 2N to divide into 2N. And our red blood cell also, and I know you know what the red blood cell means. Red blood cell in humans is called erythrocytes. Like you've known in your biology days, you've known that our red blood cell is all known as erythrocytes. And erythrocyte is responsible for what? Is responsible for transport of oxygen throughout the body. So erythrocytes, a division in erythrocytes also is the process is known as a mitotic process, or the process that we have is called mitosis. So we have mitosis in the red blood cell. And back to our last option, which is the option D, a sperm cell. A sperm cell, formation of a sperm cell is as a result of what we know as what? Meiotic division. So a meiotic division is what gives rise to a sperm cell. So the right answer to this question would be what? A sperm cell. A sperm cell is the one that undergo a meiotic process. So back to our next question. Next question, we have a genetic counselor advises couple that they could produce several embryos by IVF treatment. During IVF treatment, each fertilized egg forms an embryo by cell division. Name this type of cell division. So IVF treatment is a form of treatment that is used to help people with fertility issue. And you know, in the world today, fertility issue has been a case that most of newlywed couples just in chaos. Their home are in chaos because of what? Fertility issues. So there is a science trying to help the fertility issue through IVF treatment. And what do you mean by IVF treatment? I'll rub the board. I hope you're following what I'm trying to say. All these are what you will learn when we have the full class itself. So with IVF treatment, what do you mean by IVF? IVF is what known as in vitro fertilization. So IVF is in vitro fertilization, and in vitro fertilization is used to help people with fertility issue. So like the question asks, a genetic counselor advised couple that they can produce several embryos through IVF treatment. During the IVF treatment, each fertilized egg forms an embryo by cell division. So the IVF treatment has been used to help the couples that are not able to give birth. So the IVF treatment, we have the sperm of the husband being taken, and we have the sperm cell, like we said. We have the sperm cell, and also we have the ovum from the wife. So the sperm cell and the ovum fuses together in a place outside the uterus. This is done in the laboratory labs. IVF treatments are done in laboratory labs. So we have this coming together in the labs to give us what we call a zygote, or zygotes rather. So after the zygote has been formed, we have series of developmental process in which the zygote undergoes to form embryo. So the embryo continues to develop in the mode where it is being kept, maybe a test tube or in another medium. So if we have this zygote developing into what we know as what? Embryo. And mind you, we've previously identified that a sperm cell has a what? Haploid number of chromosomes, which is represented by N, and an ovum also has an haploid number of chromosomes, which is also represented by N. And the coming together of the sperm cell and the ovum gives us a zygote. The N and N fuses together to give us a what? 2N zygote. And 2N zygote also divides mitotically to give us a what? 2N embryo. So the process in this IVF, in vitro fertilization, the process of the self-division that we have in the zygote forming several embryos is what known as what? Mitosis. So the mitosis is the process in which the several embryos are being formed. So, alright, I'll take a short break with my Coke. So, sorry, I need to take something to hydrate myself. I know you can also do what? Join me in also relaxing yourself a bit with a bottle of chilled drink. So, in some minutes, we'll just. Alright, so that's relaxing. So, thanks. Also, please, I don't want you to take your attention away. I'm just trying to hydrate myself. Alright, back to what we are saying. In vitro fertilization. You've seen what we mean by in vitro fertilization. In vitro fertilization. Okay, so it's a serious business, students. You can see that's what you've got. You've got someone that will give you the best in what you need to know about biology. Okay, for the in vitro fertilization, we've seen, we've told you, or I've told you rather, that the sperm cell and the ovum come together to form a zygote, in which the zygote also divides mitotically into an embryo in a process called mitosis. So we now go to some questions in which you have to solve judiciously. These are questions that are asked in various examination body. It's a form of written question, but the answers are something that you must be very, very judicious about. And you know, in biology, we are more concerned about your spellings. Spellings are very, very, very important in biological questions. To have the right answers or to have the right grades in biology, you have to have a good mastery of spellings. Your spelling drill must be very, very sound. So the next question I will be having is a form of a theoretical question. We are asked, a person was involved in a car accident and became unconscious due to excessive blood loss. Explain briefly how the blood loss can be restored. So I know a person was involved in a car accident. Imagine you are a witness of a car accident, or a fatal accident has just happened in front of you, and the person was unconscious as a result of excessive blood loss. How could you help salvage the situation, or what is it as a biology student that you can do to help restore blood loss in that critical condition? So how do you go about this? I rub the board. So what you need to do in this kind of situation is not just to panic, is to, the first thing is what you have to take the victim. You take the victim to what? To nearest hospital. So you take the victim to nearest hospital, and in the hospital, to salvage this, what must be done to restore blood loss is to know the genotype of the patient or the person whose blood is being lost. So the genotype is crucial to know, to take notes, so as to avoid something that will call agglutination. Agglutination is a serious thing that can lead to the man's death. So, and after you've taken the victim to the nearest hospital and the genotype has been carried out, and the next thing, just follow, the genotype. I hope you're with me. After the genotype has been identified, so the blood group after the genotype has been identified, so the genotype gives us the blood group of the man. The genotype of the man, we have 6 genotypes. Like, we have 6 genotypes, which are your AA or AO. We have the B, BB or BO. All these are what you learn when we start our classes. We have the other genotype, which is your AB. AB does not have any other one, and we have the other genotype, which is this 1, 2, 3, 4, 5, and we have the last genotype, which is OO. So all these are the major genotypes that we have. We have the 6 genotypes. We have the AA, we have the AO, we have the BB, BO, AB, and OO. So the genotypes also gives us the identification of the blood group. So the blood group is a phenotype. The blood group is a phenotype of the genotype. So we have how many blood groups? So we have from this, you can see that our A, we have the blood groups which we can guess from our genotype. So AA or AO we have blood group A. We can only have blood group A with that. And with BB or BO, we have blood group B. And these, because A and B are dominant, they are codominant. You still learn about much of this term when we start the class fully. So A and B are codominant. So we have the blood group of AB, and we also have O. So we have how many blood groups? We have A, B, and O. We have we have blood group B, and we have blood group AB and blood group O. So after the genotype has been determined and we know the blood group, is a successful transfusion of blood can now occur. Why is this necessary? This is necessary because to avoid agglutination. Agglutination is the clumping of contrasting antigens and antibodies. So agglutination occurs when we have contrasting antibodies and antigen together. For example, a person with blood group A can only receive blood from blood group A and O. Why is it possible? Our O is a universal donor, and our AB, from your knowledge of biology, is a universal recipient. So our B also can only receive blood from blood group B and O only. So this is why it is crucial to determine the blood group of the man to avoid what we know as agglutination. So agglutination is very, very important so as to save the life of the patient that we have. So the next question we have here is we should look at the crossing. Yes, please students, I want you to take note of this, because this has been a place where students fall victims and they don't perform well in the process of answering questions like this. The questions are on genetics, so I want you to listen and pay attention as I go on with the question. I need to rub words on the board, so I hope you are with your pen and you are paying close attention to what you're trying to say. So the question that we have is, this is very important for those writing a written examination, because there are steps you need to take to answer the questions correctly. We are asked that two heterozygous yellow plants are crossed using a genetic diagram. State the phenotypic ratio and the genotypic ratio. Two heterozygous yellow plants are crossed, and we are asked to state the phenotypic ratio and the genotypic ratio. Note this word. In concept of genetics, we have what we know as homozygous, and we have a term called heterozygous. So from the word homo means the same thing. We have the same alleles, the same gene. So, need to rub my face. Alright, back to business. Homo means the same allele. For example, we can have an homozygous trait for tallness, which is T. Let's just say we are representing our tallness trait to be T. So we have homozygous, since we have the same, we have T. So we have a pair of traits. And for any heterozygous, we have contrasting allele. Contrasting allele, for example, for any heterozygous traits, heterozygous tau, we can say we have our capital T, which is the other form of this capital T is what? The smaller T. So we have this to be in homozygous condition. We have this homozygous tall plants, and this is what heterozygous tall plants. So, but look at the question. This is where students get it wrong. The question states that two heterozygous yellow plants, in the basis, talking about genetics, since we are not asked for tallness, it will be wrong for a student to represent his tallness with T, because the examiner has given a clue of what is probably the best letter to represent the traits that you are being asked. Back to the question that we asked us, we are asked that two heterozygous yellow plants, and the traits that we are discussing here is color. So the trait that we are talking about is color, and we have heterozygous yellow, Y for our yellow color, and since we said heterozygous mean another form of the traits, like I've explained earlier, for the tallness we have another form of tallness, which is the T. Basically, you can, from your knowledge of genetics, the basic knowledge, you know that T is the dominant one and the small t is the recessive form. So we can say we have the dominant form for yellow color, which is Y, and we have the recessive form, which is the small y, crossed with another heterozygous yellow plant, which is Y and Y. So this is the first stage. You have to write out the traits that you are crossing. So we have yellow, heterozygous yellow, and we have what? Heterozygous yellow plant. So also, and the next thing, this is where most of students just come across the mistakes they make. So we have, the next thing you should do is you write out what we call the gametes. We have the gametes here. The gametes is the next thing that you should write out. Some students will just go on just to cross. They cross and they get all their likes answers, but this is wrong in the sense that we're not having a multiple choice examination, but a written, for the purpose of a written examination, you have to go through the process that is necessary for you to have the correct grade. So, like I said, you have to put out the gametes, and we have just two gametes here, which is Y. So it's more beautiful when you put them in a circle to show that they are gametes, and the other gamete, the small y, also with and the other. So with this is the second step which you've taken. You've given yourself a better chance of getting the right option. So from here you get to cross. So you cross. When you do your crossing very well, when you do your crossing, we have what we know as our y crossing together with our y, and we also have the same y here. We'll cross it together with this, and when we have two y coming together, from what I explained here, you know, when you have the same traits, you have what? Homozygous. You have y. Probably I'll rub to make you see what I'm demonstrating. I'm with you. Don't worry, I just want to make you have a wider view of what you're trying to do here. So this y, and this y. Being crossed, we have y like we said, a gamete, this capital Y, and there's a small y. Also with the other gametes, this is very crucial for those writing the written exam. So you cross it, you have your Y and your Y, which is homozygous, and you cross it with the other Y. And don't forget, this is as a result of Mendel's law. Mendel's law of segregation of gene. So they segregate separately. Now, you know the first law of Mendel, genes segregate separately. So we have Y also to give us an heterozygous. So the other gene crossing with this. Please, I need you to focus on what I'm trying to do. Don't mind the drawing and the stick up and down. So you just have to focus on where I'm going. So this and Y, and you have the last one. When you cross this very well, we have the last one to be what? Y and Y. So from here you have the probability of the offspring being homozygous yellow, heterozygous yellow, or having another color which is not yellow, maybe white. Yes. So from here we are asked for the what? Genotypic ratio and the phenotypic ratio. Phenotypic ratio, phenotype is what you see physically. Phenotype is what you see, what you can observe physically, and from here we can say our phenotype we have what? We have a yellow plant. Yes, we have another yellow plant because Y is dominant over Y. I already told you the last time that the small y is recessive, and we have Y dominant over Y. We have the small y. So we have how many yellow plants? We have one. This is a yellow plant. This is a yellow plant. This is a yellow plant. So we have phenotype. We have 3 yellow plants. We have 3 yellow plants, and we have what? 1. Let's call the Y, the recessive form, to be white. 1 white. So the phenotypic ratio, you can see this is a ratio of 3 to what? 3 to 1. You can probably see from that. But talking about the genotype is the constituent, is the composition of, is the whole composition of what make up the organism. We have the genotype is talking about the all composition of the organism, and for the genotype we have what we know as the same traits, which is your homozygous. We have 1 homozygous yellow. We have homozygous yellow, and since these are what they are contrasting characters, we have what? 2 heterozygous yellow, and the other one we have one which is the white man. So we can say that the genotype or the genotypic ratio for this is in the ratio of what? You can just look at the top, the genotype. I want you to see it clearly. So the genotypic ratio, rather, we have 1:2:1. So with this, you have a good score, or you have a good grade solving the genetic question. So the last question for the day, we are just asked to just give the similarities between transpiration and sweating. As you can see, for the past few minutes now, I've been sweating profusely. So we are asked to give the similarities between transpiration and sweating. So transpiration, and I'll rub what's on the board. So I want you to take your pen and your writing materials just to get what we're about to say about transpiration and sweating. Transpiration, it occurs mainly in plants. It occurs mainly in plants, and whereby your sweating occurs in humans. But mind you, mind you, the difference between these, the key difference between this is transpiration occurs during the day, and whereby sweating you can sweat during the, imagine you going to your bed during the night and you wake up in the next morning, you find that your bed is wet as a result of what? You're not bedwetting, but you find out that or maybe the room you were is not well ventilated, and as a result of that you have to produce more sweat. So sweating occurs during the day and night, but transpiration only occurs during the day. And the similarity that you should take note, similarity that is very, very pertinent to, that is common to both sweating and transpiration, is that both brings a cooling effect. Imagine you, after you've eaten your food, you've rushed your food, imagine the amount of sweat that comes out of your body as a result of the hot food taken in. You can see that after some time you feel relieved. After taking some breeze, you feel relieved as a result of cooling. So sweating brings a cooling effect. Also to plants, transpiration brings a cooling effect. So I hope you've enjoyed yourself with this short and concise explanation on heredity, genetics and reproduction. I want you to do well to join us next time when we was going together through another concept in biology. Till next time, have a wonderful day. Thank you.